In this last lecture, we're going to look at a few conditions and how defects in enzymes can have dramatic effects on the metabolic pathways that are taking place in the cell. By the end of this lecture, you should have a really good understanding of the relationship between the pathways, as we're going to apply them to a few case studies and see what effect mutations have. For those of you that are interested in going into medicine, we'll also look at how you can use blood and urine tests to understand what's going on at the cellular level. So we'll compare and contrast the anabolic and catabolic pathways, and as always, regulation is going to be very important. There aren't many supplemental readings associated with this lecture. Based on everything that you've learned in this course, the lectures and the assignments, it really should provide you with the fundamental knowledge that allows you to understand any disease at the cellular level. And if you are going into medicine, things are going to get even more complicated. You really do have to understand the basics and then be able to apply them to understand what's going on in the tissue and then the organ level, as well as the overall implications for health in the body. So I really hate to be that annoying person that says we're going to circle back to something, but we really are going to come back to this map, and you could even draw out your own summary so that you can understand the relationships between the pathways. If I was in my office right now, I'd show you some of the maps that some students have done in the past, but really it's not about memorizing all of the individual steps, but really understanding the relationship between the pathways. So where the connections are made and what key enzymes are regulated and what they do. I have included protein breakdown and the urea cycle on this slide, but remember, this really is the last thing you want happening since proteins play essential roles in the cell. And even though we're not really going to cover the urea cycle, it is a catabolic pathway that's important for getting rid of any excess nitrogen found in those amines that are present in amino acids when they're broken down. When it comes to metabolic diseases, there are some that are tested at the genetic level when you're born, and these would be called inborn errors of metabolism. And PKU or phenylketonuria is one that you may have covered in your biology classes. It really comes down to the function of an enzyme and how it's regulated. So remember, these can be either inhibited or stimulated in a variety of different ways. And defects may arise either due to mutations in their primary sequence or even due to a lack of gene expression. So if there's no mutation detected in the gene, it could be due to a mutation in another protein that's needed for its production. We can use the levels of different metabolites in the blood or urine to help us understand where the defect lies. But there are many considerations that we need to take into account, such as the stability or the variability in the molecule. So it might be metabolized or processed, so we might actually have to measure different compounds. And then it also comes down to what is a significant change. Are you looking for 2 times or 10 times more than your controls? The pathways are very complex, so much more so than what I've provided in my cartoon. So do keep that in mind, that the results might not necessarily tell you what's going on. But it can help allow us to come up with treatment options in order to bypass the defects and maintain sufficient energy levels or produce necessary molecules that the cell is going to need. So let's look at some case studies. Rhabdomyolysis is a syndrome that occurs whenever there's damage to your skeletal muscle. Rhabdo just means rods or wands, and this refers to the structure of the cells and structure of the muscle cells, or the myo part, and lysis just means breaking. So whenever these muscle cells are damaged, they can release their intracellular components into the bloodstream, and clinical biochemists and doctors can use these different levels of molecules in the blood and urine to deduce what tissues and organs have been affected. Creatine kinase was that enzyme that helped produce ATP from creatine phosphate, and if this enzyme is found in the serum of the blood, this indicates that muscle cells have been damaged. And elevated levels of creatine kinase can actually indicate that rhabdomyolysis has occurred. And one of the important muscles that may be damaged is the heart. So if there is a blockage in the blood vessels supplying oxygen to your heart, those cells won't be able to make ATP and will die, releasing their molecules into the bloodstream, and creatine kinase is going to be one of them. Another abundant molecule that may be released is myoglobin, and remember, this protein is specific for muscle cells as well, and when it's released, the heme molecule gets metabolized and can actually turn your urine a brown color, leading to myoglobinuria. So on the right is Uncle Rhabdo, and I'm not sure if there's any CrossFitters out there, but CrossFit is a type of extreme exercise. And this used to be the mascot, where you can see that he has some muscle damage, which may be due to hypoxia or not supplying your cells with enough oxygen. This is also known as ischemia. But it can also lead to kidney failure, where we can see that he's hooked up to a dialysis machine because there's lots of cellular metabolites that are spilling into the bloodstream, and your kidneys are just trying to filter them all out and get rid of them. So rhabdomyolysis can also occur under temperature extremes, so providing that energy to disrupt all the non-covalent interactions between molecules, or at low temps, preventing the movement between molecules. And we can also see rhabdomyolysis in the presence of different drugs or infection. And as we'll see in the next slide, in the case of metabolic disorders where you have a mutation in an enzyme that's important for metabolism. So one of the metabolic conditions where rhabdomyolysis is seen is McArdle disease. There are a bunch of different glycogen storage diseases, and McArdle disease arises when you can't break down glycogen in muscle cells. So it is classified as a type 5 glycogen storage disease, or GSD for short. There are about 65 different mutations in the gene encoding for glycogen myophosphorylase, and this is the enzyme that's needed to remove glucose residues from the non-reducing ends of glycogen. So in the presence of these mutations, glycogen can't be broken down in muscle cells, so they won't have access to any of the energy that can be made from this glucose 1-phosphate. We look at it in the context of the pathways. We're essentially blocking this step in muscle cells specifically, but we can still get energy from other molecules other than muscle glycogen. So I want you to think about the relationship of the pathways and answer the following questions. I am going to walk through these answers, but it really is important for your own learning that you attempt to answer these questions based on what you've learned about the metabolic pathways. Let's start by understanding the biochemical basis of some of the symptoms of this condition. So do you really understand the biochemical basis of why they might be experiencing muscle fatigue and weakness as well as rhabdomyolysis? Pause the lecture and think about the pathways in a muscle cell, and then we'll go through to the answer. So the most obvious answer would be that since they can't break down glycogen in muscle cells specifically, they won't be producing glucose for glycolysis and won't have access to this amount of ATP. But there's actually another reason why. If their muscle cells don't have access to this glucose, remember pyruvate that's produced in glycolysis can be converted to oxaloacetate, which is needed by the citric acid cycle to run and get energy out of other molecules. Just moving on to the next point. So one thing that these patients may experience is a second wind phenomenon when exercising. So they may be initially tired, but after a while, a burst of energy will kick in. Do you know why biochemically this may occur? Pause the lecture and think about why this might be happening. The release of epinephrine and maybe even glucagon, if all the blood glucose is being used up, will signal for the breakdown of fats and liver glycogen. And your liver may also be producing glucose in gluconeogenesis. So this can provide muscle cells with fatty acid chains as well as glucose in order to efficiently make ATP from all of that acetyl-CoA. Now, what suggestions would you have for someone that is living with this condition? So just related back to the last point, you might suggest that they do a slow warm-up when they're exercising, and exercise will be very important in their condition for a variety of reasons. But this will really allow them for that fat mobilization to take place as well as liver glycogen breakdown in order to supply that energy to their muscle cells. When it comes to their diet, or even any of our diets for that matter, complex carbohydrates are always better since it does take a longer time for them to be digested, so you won't see this huge increase in blood glucose. The cells can then slowly take up that glucose and use it for energy. Now, what are some other complications you could foresee for these patients? Some things that we might be concerned about for somebody who has McArdle's disease is hyperglycemia. So if you have hyperglycemia, this can lead to type 2 diabetes. So this is because their muscle cells can store glycogen, but they can't break it down. So effectively, all of their glycogen stores are filled up with glucose, and they can't store any excess glucose as muscle glycogen. Okay, so it is a better idea to have those complex carbs that slowly get released and used throughout their body. If you were to hit their body with a ton of simple sugars, some of them will get used, but then we won't be able to use the rest of them, and that's what can lead to hyperglycemia. Another condition that might arise is ketoacidosis, which we looked at in the last lecture. So because they are relying primarily on fat breakdown, in the absence of glucose in supplementing that citric acid cycle, you can get the buildup of ketone bodies, leading to an acidotic state. The other thing to keep in mind is if we can't store any excess glucose as muscle glycogen, we could store them in fats, and this could lead to the synthesis of lots of VLDL particles, leading to cardiovascular disease. And then the last idea or concept we haven't really covered, but if they are breaking down proteins for energy because they can't use muscle glycogen, then this will increase the nitrogen load, and in order for the urea cycle to remove it from the body. Another condition that can lead to rhabdomyolysis is due to a deficiency in carnitine palmitoyltransferase 2. Remember, in order for fats to be broken down, they do need to be imported into the mitochondria, and this involves the carnitine shuttle. There are different transferases depending on the length of the fatty acid chain, and carnitine palmitoyltransferase 1 will transfer 16 carbon chains onto carnitine for import by the translocase. And then carnitine palmitoyltransferase 2 will transfer it back onto coenzyme A. So if you have a defect in CP2, the 16 carbon chain will be stuck on carnitine, and we won't be able to break it down to acetyl-CoA in beta oxidation. So let's think about the implications for the metabolic pathways in this condition. Take a minute and think about what some of the complications might be for someone with a CP2 deficiency. So the obvious one would be not having access to ATP from the breakdown of 16 carbon chains. And because we can't break down fats, they will be primarily relying on glucose, so hypoglycemia may arise. The muscle fatigue and weakness will also occur along with rhabdo if there is extreme damage, and kidney issues might also arise due to the renal excretion of the metabolites that are released into the bloodstream. You can live with a CP2 deficiency, so how do you think cells might compensate in order to still produce energy? Since we are heavily relying on the breakdown of carbohydrates, you will see an increase in the level of enzymes needed for glycolysis as well as creatine kinase in muscle cells. Phosphocreatine is a limited storage form of phosphate, so it can only help out a little bit, and that's why we won't necessarily see a huge increase in creatine kinase in muscle cells. Now, what conditions can you think of that could worsen these symptoms for these patients? Pause the lecture and think about this question. If someone was fasting or exercising, they will be in the catabolic state and will be trying to break down fats and glycogen in order to produce energy. If they can't get access to any energy from 16 carbon chains, then this could aggravate or lead to symptoms. Also, if they were consuming lots of fats and not a lot of carbohydrates, then they won't be able to get energy from the 16-0 chains from their diet, and they also won't be filling up their glycogen stores. So what suggestions would you have for someone that's dealing with this deficiency? Since we know that they are primarily relying on carbohydrates for energy, a diet consisting of 70% carbs and proteins can allow for efficient energy production. And in this case, carnitine supplements can help since they will be tied up with any 16 carbon chains that are present. So instead, medium chain fatty acids can be given and broken down for energy, but only if free carnitine is available for the carnitine shuttle. The last thing I want to mention is lactic acidosis, which can also arise in metabolic diseases. Whenever you have a block in the pathway, it is going to result in metabolites being shuttled into other reactions. We saw that a lack of oxygen could lead to lactate production, but there could also be a mutation or a lack of cofactors for a downstream enzymatic reaction that will lead to pyruvate being converted to lactate. And it is indeed lactate that's made in this reaction catalyzed by lactate dehydrogenase. Now, it was initially called lactic acidosis because there is a lactate proton cotransporter that exports the lactate and a proton out of the cell. But the source of the protons is actually from ATP hydrolysis or any chemical reactions within all of the catabolic pathways. So acidosis is a common symptom that is seen in a lot of the metabolic diseases, and by this point in the course, I really do hope you have an appreciation for the importance of pH at the biochemical level. The metabolic flux really dictates how molecules flow through the different pathways. We've seen how some steps are essentially irreversible, while others are reversible and depend on the concentration of metabolites to dictate the directionality of a reaction. It's the regulation of these irreversible steps that can divert molecules to other pathways. And we've seen how signaling can turn on and off the catabolic and anabolic pathways through a variety of mechanisms. Whenever you do have a block in the pathway, it really comes down to understanding the relationship between the pathways in order to wrap your head around what's going on in the entire metabolic network. There are lots of websites as well as maps that you can access online if you ever want to look up a disease, and then you can try to figure out what effect it will have on a cell. So my final key messages are really for you to understand the relationship between the metabolic pathways. And remember, it does depend on what cell type you're looking at because the cellular context will really be important when considering what's going on for the rest of the body at the organ level as well as at the organism level. Metabolism is really important when it comes to ATP production from the catabolic pathways, and then that'll generate energy as well as intermediate metabolites that can be used to synthesize macromolecules in our anabolic pathways. There's going to be lots of proteins and enzymes that are involved, and many of these will involve and require vitamins or other mineral cofactors for their structure and their function. Hormone signaling was really essential in order to regulate and turn on and off these pathways, and this will allow different cells to use the different metabolites either for energy or for the production of larger macromolecules. So keep in mind that biochemistry really is everywhere around you, and so I really want you to share what you've learned and keep asking questions about what's happening at the molecular level. If you can understand what's going on at the cellular level, this could particularly save your life or someone else's if you are experiencing any disruption in the metabolic pathways. So I do want to leave you with the thinker question. So what kind of ideas and skills are you going to take away from this course? So this is an online course, so you really had to work at working independently and doing some research in order to come up with some answers. But how can you apply what you've learned either through those skills or the knowledge of biochemistry to your own health, as well as your overall career goals and maybe your program choices? There will be brain break questions posted on Quarkus for this lecture, but I would also ask if you could kindly fill out the course eval so that we can evaluate how this course ran in order to improve for future offerings. Thanks for sharing this journey into the wonderful world of proteins with me and for inviting me into your self-isolation worlds. If you're interested in learning more about proteins, check out our website and all the other courses that are offered, and they can be used to fulfill your degree requirements. Do take care and let me know if you have any questions about the material or your options when it comes to all that biochemistry has to offer.